Excuse me? Okay. So stuff about the project is the poster project is up on the piazza, and it's also explained on the blackboard, so you need to have, all of the group members need to go and look at that information so you understand exactly what the project is and what the milestones are. But today we're going to go ahead and finish membrane transport. And we were talking about the sodium glucose transporter and glucose absorption as a model, but we sort of left out one of our secondary active transporters. And so obviously if you can have sym ports, you can have antiports. And our poster child for that is the sodium calcium antiport. Okay? And it's found on many different kinds of cells. And one of the classic cell types that it's located on is in the cardiac muscle cell. So let's draw a diagram of how that works. Okay. And so if you have a cardiac muscle cell, the sodium calcium, it can be called the sodium calcium antiport. It can be called the sodium calcium exchanger. So there's a couple of different synonyms, but it's actually, it's going to be running off the electrochemical gradient of one of those ions, and it's almost always sodium. And then the other one is moving against its electrochemical gradient, that's almost always calcium. So if we put this on the cell, and what happens is, in order to pump out calcium, we want to always keep calcium low in between muscle contractions. And it's actually in a cardiac myocyte, it's calcium that carries the action potential. Okay, that's what carries the depolarization wave. So calcium in all cells is highly regulated, so we tend to want to really clean it out of the cell and before we have some sort of signaling faults. So to do that, we have to eject it, and the calcium level outside the cell is 1,000-fold higher than it is inside the cell. In fact, the numbers are, it's about 2 millimolar outside, and inside the cell it's 0.1 micromolar. So it's 100 nanomolar inside the cell. So this huge, it's going against a huge chemical potential difference. And also the outside of the cell is more positive, right, by 60 millivolts. So it's going against both an electrical gradient and a chemical gradient. Okay, so that takes a lot of energy. In order to be able to move this transporter against that kind of gradient, we need to take 3 sodiums, okay? And the energy that's expended is a direct function. It's actually exponentially related to the number of sodium ions coming in. So that actually gives us enough power to move 1 calcium ion out of the cell. Now, one of the questions when you're looking at all of these transport processes, you should ask yourself, whether it's a pump or an exchanger or a sym port, is does the cycle that this goes through, does it cause a net movement of charge? That is it electrogenic? Here's that word. Does it generate a current? Okay? And so the way you do that is you just need to see if there's a net movement of charge. And so in this case, we have 3 sodiums coming in, and we have 1 calcium moving out, but it's a valence of +2. So we got 3 pluses going in, 2 pluses going out. So the net is 1 plus charge for every cycle that this thing goes through the cycle left inside the cell. And a really quick little equation to keep inside your head so you don't get messed up is just in minus out. That equals the net charge that transporter is producing inside the cell. So in minus out. So quickly you just go 3 minus 2, right? Because it's 3 pluses coming in minus 2 pluses going. I'm sorry, yeah, 3 pluses coming in, 1 calcium, that is 2 charges going out. So 3 minus 2 is 1 positive charge left in. So this is actually, and you also need to, so if you leave extra charge inside the cell, there's a name for that. It's going to cause the membrane potential to become more positive, and when that happens, it's called depolarization. So we're depolarizing the cell. So if I'm asking you, does this transporter hyperpolarize or depolarize the cell? Your answer is based on the type of charge left inside the cell. Now, we've done an example with the sodium pump. Quickly tell me, what does the operation of the sodium pump do to the membrane potential? Does it depolarize or does it hyperpolarize the cell? Just do this math. And so it leaves what charge after the operation of one complete cycle is left in the cell. So it's +2 coming in minus +3 going out, -1, right? So that hyperpolarizes the plasma membrane. So the operation of that pump is actually going to make the membrane potential steeper. Depolarizing it makes it less negative. Okay? And here's another thing that you can associate with those two phenomena. If a cell is becoming hyperpolarized, it's inhibited. Whether it's muscle contraction or neurosecretion or hormone secretion, it's inhibited when it's hyperpolarized. If you depolarize a cell, it tends to become active. And that's particularly true for muscle cells, like the cardiac myocyte. So it's more likely to undergo contraction if it's depolarized. Okay. All right, so now let's test our knowledge of putting things together. So that's going to be, we're going to briefly go back to the little epithelial examples so we know how to combine these, because these transporters never operate by themselves. They're always working in concert with something else. In fact, this thing can't do its job unless you've got a sodium pump that's taking sodium out and keeping the concentration of sodium low. Because every time the sodium comes in, it's going to tend to raise the concentration of sodium. And so we need to eject the sodium to keep it low. And that's exactly why you have a sodium pump on every single cell. Okay? So there's the sodium pump. Now, so a question that's asked in the outline is, if you give a patient Wavane, and you'll actually give them a drug called Digitalis, but they do the same thing. They temporarily block a fraction of the sodium pumps. Why would you give that to a patient that has cardiac insufficiency? So their heart is not beating hard enough with enough force to deliver enough blood throughout the body. So you want to increase the contraction strength and then the number of contractions per minute. Both of those will increase the cardiac output of that patient, and so this is something that will strengthen that person's heart contraction. How is that going to work? What you're doing is you're blocking the sodium pump. How's that going to work? So think about it by yourself. How does that produce a more strong contraction? Draw it out. Think about it in your head. It's better to draw it. In fact, I encourage you to do that with all the exam questions. We'll try to do it up here. I'm running into several people that are making mistakes in their head, but if they had it on paper, they wouldn't make those mistakes. Okay. Now, get with your group and come up with a collective answer. That's called you gotta try. Well, that I think so it has to become that'll increase the sodium concentration, and that'll prevent sodium dioxide and that'll concentration of what is it the cell low calcium. All right, so I'm thinking of a number between 1 and 18, 17. What's your answer to this? Why is that person's heart going to contract harder? There's no such thing as a ridiculous answer, but try to make it. You have an idea? We're 17. Raise your hand collectively. You're what? You're over here? Okay. Do you have a victim? I mean, a spokesman? Okay. What do you think? Just what makes you think that that's, or what could be reasonably causing a person's heart to contract harder? One thing you need to know is something a little bit about muscle contraction. What stimulates muscle contraction? Calcium. Pulse of calcium. Okay. But we need to have a pulse and we need to kick it out. So we want to have more pulses of calcium, right? Okay. What else can you do to make it? You can actually make that person's heart contract harder if you increase the concentration of calcium per pulse. Okay. So you want something to happen up here that causes the intracellular calcium to go up briefly. And how is Wabi going to do that? So the logical, so anybody else want to try? I guess we're done.
Well, you basically did the answer for me, but
pardon me?
I think you've already answered the question, but if you're shutting down some of the sodium pumps, then you're going to cause an increase in the amount of calcium in the cell.
How?
Because you can't exhaust the sodium.
What's good? So that's the way to answer this question is follow it from where the drug starts, right? And you're doing that, I think, because you sort of lead to the answer. So you want to go here to go. What's going to happen if you block this to potassium and sodium? But you don't really care about potassium because there's no link between this and that. It's in terms of potassium. What's the common ion?
So you're going to have too much sodium in the cell.
It's going to go up, right?
Right, and then that's going to cause some trouble with the simpler.
Because that's the energy that transporter is running off of. It's running off of the power of sodium to come in the cell and cause this thing to go through its cycle. And so if this thing is blocked, then what's going to happen to the calcium level? It's going to go up, and that person's heart will contract harder. Okay, so it's not a complicated thing, but you have to do it logically, and don't try to just jump to the answer. Just follow the path of the inhibitor, and through how are the two mechanisms related? And that's how you answer those kinds of questions. Okay. This is? Oh, it's on. Okay, good. But the sound is no way. Okay.
We'll get in there afterwards.
Okay. Thanks, Troy.
You're welcome.
Okay, so let's just review. So now we're going to put some stuff together. And we talked a little bit about this, but I want to make sure that you got all the players. Let's do it this way. Let's turn Bart Simpson on his side. Okay, so this is an epithelial cell, and then we're going to connect it to another epithelial cell like this. And so this is going to be the lumen over here. Okay, so food is going up and down this way, and then the blood is over here. Okay, so if you eat a candy bar, and so you're going to ingest some sucrose, how are you going to absorb that into the bloodstream? Sucrose will not go across the epithelium. How are you going to deal with it? Well, what is sucrose?
Sugar.
It's a disaccharide. Okay, it's composed of two sugar groups, two small groups, glucose and fructose. So what you need some single-spanning membrane protein that is sucrase. That's a digestive enzyme, and that splits sucrose into glucose plus fructose. Okay, Now, so we're on the luminal side, but we want to go to the blood. So this is transepithelial transport. That means there's two membranes we have to go across. Transepithelial transport. So how do we get the glucose to go across? And let's say this has been a while since you've eaten that candy bar, and this cell is not in your duodenum. It's not in your jejunum. It's in your ileum, right before it goes into the colon. So this is the last chance to get a little bit of this sucrose, or not the sucrose, but the glucose and fructose absorbed before it leaves the small intestine. So the concentrations are low. They're in fact much lower than the glucose concentrations in the blood. So we'll put this way down here to indicate that this is low in concentration and that the blood concentrations could be 5 millimolar. Okay, something like that. How are you going to get it across? Well, you can tell, you look here to here, you have to invest energy to pump something uphill. So you know there must be an active transport step somewhere. But how do you move glucose across membranes? You already know two mechanisms now. Pardon me? That's Synport, which is called, what's the nickname? You don't want to call it the sodium glucose cotransporter, so you call it SGLT1, right? All right, so we're going to have SGLT1. And you don't want 2 because SGLT2 is in the kidney and it's got a low affinity. So you want this high affinity. So this is going to mop up every bit of glucose. And so it will pump glucose up to a high concentration in the cytoplasm. And it will bring in sodium. And it doesn't bring in one sodium. The stoichiometry you need to know, it's 2. 2 per cycle. Right? So 2 sodiums have to load into a binding site and 1 glucose. And when all 3 of those conditions are met, then it changes its shape and flips to the inside and releases all of it into the cell. Right? Is this pump, or is this is a secondary active mechanism, is this electrogenic? This should be really quick. There's no counter ion. So you got 2 going in and nothing coming out. So it's 2 minus 0. This is depolarizing the membrane. Right? Okay. Good. So always think about, is this depolarized? What's it doing to the membrane potential? And there's 2 membrane potentials. There's a membrane potential across the apical membrane and 1 across the basolateral. Okay. All right, so we're going to build up high enough concentration so that it can run downhill through another thing. What else takes glucose that's in the small intestine? It's not a secondary active transport. We'll put it right there. Anybody? GLUT2, good, okay. So GLUT2 is our sloppy, low affinity because it doesn't need to be good. And there's another reason why it needs to have a low affinity. It needs to be not very choosy. So when you have a low affinity, it means you're not choosy, right? So glucose is going to go down its electrochemical gradient. Actually, since glucose is uncharged, it's just going down its chemical gradient. Is this electrogenic? No, glucose doesn't carry a charge, right? So you got transepithelial glucose going into your blood. Now, this will not operate for very long because why? If these are the only 3 transporters that are moving, what's going to happen? What's going to prevent it? Why is it eventually going to just go out? Yeah, the sodium is going to go like this, like that, and that's going to block this, right? But what do you have? I'm sorry.
Sodium potassium pump.
The sodium potassium pump. So where do you have to put it though? Let's do that. Let's put it on the lumen. Okay. What's going to happen then is sodium. It's going to do exactly what it needs to. Sodium's going to go out. Potassium's going to go in, and that's going to work. That will keep the sodium, and we will have really good glucose transport. What we won't have is sodium transport going across the membrane. And the reason we need that is to absorb water into the GI tract so it doesn't go into the colon. Okay? So you need to put, this will operate and run glucose transport, but it will not run fluid transport. So we want to put it, that's the reason we put it there, so that the sodium is coming into the cell and then going out through this pump, right? 3 sodiums, and potassium is coming in. That's going to osmotically draw water into your bloodstream to keep you nicely balanced. Good. So it matters where you put the pump. You might have the right pump in your head or transport, but if you put it on the wrong membrane, you'll mess up. What if we had put this thing on the blood side and we put GLUT2 over here? Why would that not work? Well, what's going to happen is well, it'll cause diarrhea. Let's call it what it is. Dysentery is a different problem. But what's going to happen is it doesn't have very high affinity, so when glucose gets below the concentration in the blood, it can't pull it in anymore. That leaves a significant amount of glucose in the lumen, and you get diarrhea. Okay. Now, there's another thing we need to remember about this thing. This will also move galactose. This is very specific. SGLT1 is very specific for either galactose or glucose. And galactose is important because that's in milk sugar. Lactose breaks down to lactose or galactose and glucose. So if the problem is reworded to become lactose, then you have to move glucose and galactose. But they will both go through SGLT2. How does it get out? Galactose will also go through GLUT2. That's why we need a sloppy pump or sloppy transporter. And in fact, it will actually take all 3 components of lactose and sucrose, because we haven't talked about this one yet. We have fructose. So what are you going to put on the apical membrane that transports fructose? GLUT5, yeah, right? So GLUT5, and it can't transport fructose uphill. There's just no way, right? So it's going to go into the cell, and it will go downhill, and how it exits is through GLUT2. So GLUT2 is so sloppy, it will take glucose, galactose, and fructose. But GLUT5 is not sloppy. It has very high affinity for one thing, fructose. And so it's truly not a glucose transporter, it's a fructose transporter. It just has that name, because it looks like it. Okay? All right, any questions about how that works? Good. All right. Okay, let's see. Let's go to PowerPoint, and at least I can see it here. We've talked about that, lecture 10 questions. So we need to, these are the lecture 10 learning objectives. So we want to identify the major differences in solute transport through ion channels versus all other transport. What are the characteristics? What kind of solutes? What kind of kinetics? What kind of protein? That's what we need to know to answer that question. We need to, there's lots of different ion channels. They fall into the category of facilitated diffusion. Okay, so categorize different ion channels into different types. And then one of those types we're going to look at very specifically, and that's a potassium channel. And we're going to look at a bacterial channel that actually is the ancestor of our voltage-gated K channel that's used in neurons in a lot of different places. Okay, and then finally, we're going to answer the question is, if you have 2 competing kinds of energy, in other words, if you have potassium that's high inside the cell and it's low outside the cell, you have a chemical gradient directed to go out, and then it's positively charged, the membrane potential is positive outside and negative inside, right? That's going to oppose the movement of potassium. So when you open the ion channel, which way does it move? You can't answer that question unless you know how to use this equation. And so we're going to learn how to do that. Okay. All right, so here we go. The there's several different kinds of ion channels, and the types of solutes that go through ion channels are almost exclusively ions. Okay, there's rare differences. So the type of solute is an ion. Okay, the energy is always passive, so there's always facilitated diffusion. And remember, a channel protein is not a transporter. It's different. It's very different. The only thing similar between transporters and ion channels is that they always ion channels always contain several transmembrane alpha helices, just like transporters do. So they have that in common. But the mechanism is very different. And finally, the kinetics cannot be described by the Michaelis-Menten equation. Okay, and we'll point that out. And we won't go into the equation that actually does describe it because that's a physiology course in itself. The kinetics is very fast. 10 to the 5th to 10 to the 8th ions per second. That's the fastest transport process we're ever going to look at. So it's very fast. And generally very specific. Okay. Now, this shows the different kinds, but there's a really nice website at the University of Toronto that I prefer going to show this. And so we'll use this website a couple of times today. I think I already have it up. We're going to have to... Okay. I'll put this link. I think it's actually up already in your... but if it's not, I'll put it up on Blackboard. This is a really good way to look at ion channels. Let's just go up here. Okay, so the way they divide ion channels up are specific ion channels and non-specific. We don't really go into that distinction, but there's several names for broadly dividing them into two big groups, and one is they call it passive. I call it non-gated or resting or always open. So passive, open, and non-gated is the same thing. It means it's usually open most of the time, and so there's nothing controlling the gate. All right? As opposed to something that's gated, that means there's something swinging the door open or closed depending on what's happening. And so what we want to ask is, in this situation, what are the different types of gating? What closes and opens the gate or the ion channel? Because ions can't move through it if the gate is closed, right? This thing is this guy is always open, so ions are always... This is also called the leak channel. Passive channel, leak channel, okay. And there's an important role for those. Okay, so here's ligand-gated channel. So there's some ligand that binds to that protein and opens the channel. And that ligand can be on the extracellular side, like a neurotransmitter. We'll talk about one of those today. Or the ligand can bind from the cytoplasmic side. Your book calls those two different types. They're the same kind of channel. They're called ligand-gated ion channels. Okay? So look at that again. Get the neurotransmitter binds. The thing changes shape. It allows whatever ions to move based on the gradients and the selectivity of that channel. Okay, so those are the two things to determine what goes through: selectivity and the electrochemical gradient. Okay, let's go to, here's voltage gated. All right, so what's happening is there's a membrane potential. Okay, this is the normal one, right? This is inside negative, that's outside positive. This is like minus 60 or 70. If this is depolarized, now we're using that term, what does depolarized mean? Well, you know what that means. It means the membrane potential becomes more positive. So there's going to be positive charges start to move in here, and that's going to cause these little protein pieces to move upwards and open the channel. And so, let's try it again. So the membrane potential is pushing on some part of that protein to open the channel. So that's voltage-gated ion channels. Voltage is the gating mechanism. Okay, and then in your inner ear, you have a mechanically gated ion channel. And the reason that you hear sound is because that sound energy is actually transmitted into your cochlea, where the hair cells are. And those hair cells detect changes in pressure. that deforms the membrane, and the mechanical deformation of the membrane opens the channel. So here's a mechanically gated channel. See? So that would be like a sound coming from your iPod or something is going to open that thing. And so it's actually causing a deformation in the membrane, which pulls on the protein, because the protein and the membrane are very intimately related. And so it opens the channel, and then the ion will move depending on the two things: selectivity of that ion channel and the electrochemical gradient for that ion. Okay? And then they come up, there's a third or fourth class that's actually quite true, and it's not quite the same as a ligand, but it has to be, the channel is going to be phosphorylated on the cytoplasmic domain by some kinase. And that happens all the time. Some kinase phosphorylates, in particular, voltage-gated calcium channels and opens them, or not voltage-gated, but phosphorylated gated channels. So phosphorylation of the cytoplasmic domain can open the channel. So you got all kinds of cool ways of fooling with the conformation to cause the gate to open. Okay. All right, let's put that away for a minute. Any questions about the gating type before we go on? Yeah.
Are there channels that are like the opposite, where they're normally open, and if they're phosphorylated, they close?
There are some. Yeah, so you can have all permutations of that same plant. Sometimes phosphorylation is good. It just depends on how the phosphorylation moves the gate. If it opens the gate, it's going to open. If it closes the gate, it will close it. So that's a good point. Because phosphorylation is not always positive. It can turn things off as well. It depends. All right, let's put this down. We'll come back to it. All right, yeah. Okay, this is just we've been able to use the Michaelis-Menten kinetics to describe to measure the kinetics for all the transporters except this. We cannot use that for ion channels. And I'll get to an equation where you can use it if you take a physiology class. Okay. All right. So we've identified the major kinds. We've categorized the ion channels and the type. And so now we need to really zoom in and look at the structure of a particular channel. Okay. And so this is the bacterial potassium channel. The structure is it has four subunits. Four subunits. This is one subunit. Okay? And so you notice that there are actually two transmembrane alpha helices. They're called, this is called S5 for segment 5. I don't know why they call it M5 for membrane. I don't know. It's called a segment. So it's S5, and the inner helix, it's labeled inner helix, is called S6. So S5 and S6. And then it's got a little, what's called a pore helix that contains this little loop. Okay? And this so-called selectivity loop, all four of these create, they line the pore. So they create the selectivity filter that decides who goes through and who does not go through. So that's the decision maker. And it's a little pore helix. So this is not a transmembrane alpha helix. This is actually a charged pore helix that's in the aqueous pore. Okay? It doesn't have to be completely hydrophilic, but a lot of it is. And it actually has a little dipole moment, so that this little part here pointing into this so-called vestibule is negatively charged. Okay? And notice, if you look at this, so this is a subunit, that's a subunit, and then they've taken away the other two subunits so you can look inside. So the other subunit would be on this side, on the back side of this, and then the fourth subunit would be blocking your view. So they've taken the back one and the front one off and tossed it away so you can see how things go on the inside. Okay. All right, now, so this is the pore, and notice that there are amino acids that are not part of the helix that are negatively charged. And you have other amino acid R groups facing the outside that are also negatively charged. That makes this a cation selective channel. That makes it choose plus charges over minus charged ions. Okay, so this thing excludes anions because of those residues. All right, so now it's going to take cations, but which cation? Does it take all of them? No, it only takes potassium. It's actually quite capable of selecting between a potassium and a sodium by 10,000-fold affinity for the potassium ion. It's extremely, exquisitely selective for potassiums to move through here. All right, so how does this work? Potassiums in solution are not all by themselves. They're hydrated by 4 water molecules minimum, okay? And what's happening is, you know, think of a picture of water in your mind. You have an oxygen and 2 hydrogens. That oxygen, it has a partial delta negative on it. It has a partial negative charge, okay? And so that partial negative charge on the oxygen is attracted to the potassium. And so there are 4 water molecules hanging on to this hydrated potassium ion. So this is, it's not small like this, it's big and bulky. And that's okay, because this is big enough to go through the pore into this inner part that's called the vestibule. And the vestibule is the last place where you can ditch the water molecules and completely become dehydrated. Because you cannot fit into this little selectivity filter unless all the water molecules are stripped off. The problem with that is that creates a lot of, you lose that energy of hydration, and so you need to replace that to make that transition energetically favorable. Okay, so we're going to deal with that. All right, so what actually gets it to go into this little tiny place is the pore helix. It's got a delta negative that even when it loses the oxygens of the water, it's actually being beckoned into this little position here. Okay, so now, why is this energetically favorable? Well, notice there's a really conserved sequence. It's glycine, tyrosine, and glycine. But notice it's not the R group that's projecting into the pore. What is that? What is this? They're all oxygens.
From the backbone.
It's from the backbone. Yeah, that's the carbonyl peptide backbone. It's not the R groups. There's nothing to do with this. Okay? And so these oxygens are perfectly placed to replace the oxygens that you just lost from water. All right? So what's going to happen is it's going to move into the selectivity filter, and then it's going to move forward. Okay? The direction is controlled by the electrochemical gradient for potassium. If it's directed outwards, potassium will leak out. If it's directed inwards, it will come in. This is a unidirectional ion channel. Most of them are. Okay? So let's say the potassium is high on the inside of a bacterial cell and it's high on the inside of us. So when you open this channel, potassium's probably going to leak. Okay? And what happens is, this is really cool, this is single file diffusion. It's one at a time, and when this guy is loaded here, another one jumps in here, and their mutual repulsion, the positive charges, shoots them out the other side. It's really cool. So it just squirts out extremely fast, 10 to the 8th per second. Okay, now, what about the energy thing? Okay, so this is why this channel is exquisitely selective for potassium. So here's the hydrated ion as it enters the vestibule. It's all cloaked there. Okay, and then it's going to be stripped off as it enters the selectivity filter, and it's going to lose all those 4 oxygens. But look what happens as it enters the selectivity filter. It's completely coordinated by those 4 carbonyls. And so this is very energetically favorable. All right? Now, potassium, actually, the ionic radius of potassium is smaller than K, than potassium. Ionic radius of sodium is actually smaller than potassium. And it also carries 4 water molecules. Okay, so why doesn't it go through here? Well, it's so small that when it becomes dehydrated, it can only make bonds with 2 of those carbonyls, and this is very energetically unfavorable because you're not losing 4 and gaining 4, you're losing 4 and you're gaining 2. Sodium will not go through this channel. That's the basis of the selectivity. Okay, and notice it's a loop. It's a loop that's in there. There's no secondary structure, but it's very rigid because of this pore helix that holds it perfectly positioned. Okay, so where's the gate? This is weakly gated, so this is almost a resting channel, but it is gated by the inner helix. So that inner helix is capable of opening and then closing. And so movements of the inner helix are what gates this channel. Okay. Now, it's not gated by voltage or anything, so the things we talked about. It just every once in a while it feels like it needs to close up. Okay, and people are still working on this mechanism. Okay. All right, so are we clear on the details of this one ion channel? Okay. Now, if it's just a bacterial channel, we wouldn't be that interested in it. Okay, I'm going to up this because I see people nodding off. All right, and so let's finish this one other thing, then we'll take a break. All right, so this is the structure, partial structure of a voltage-gated potassium channel that is the reason that your brain is working, at least some of it. Okay. All right. So this helps generate the action potential that is a nerve impulse. Okay. And so it's a voltage-gated. This is actually a voltage-gated potassium channel, and it's actually evolutionarily based on you can see the structure S5 and S6. That comes from the bacterial channel, and so this one actually has 6 segments or transmembrane alpha helices. So it's still got 4 subunits. So it follows that same plan, okay? But each one of them have, instead of 2, they have 6 segments. 1, 2, 3, 4, 5, 6, 7, 6. And notice what the other difference is. What's holding the selectivity loop? The 2-strand beta sheet. Right? So it's not a pore helix, it's a pore sheet, beta sheet, okay? But it's real minute. So here's the selectivity loop, and guess what? It's going to project those carbonyls, and this is just one of them, so you're going to have to see 4 of these guys organized around. So this is what it looks like. 2 of them are cut away, those 2 facing you, and so they're all arranged like this pore, and so this is the selectivity filter comes around and forms that selectivity filter. So things are going to come up through here and out. Now, this is gated by voltage, so we have to answer the question, let's see. So let's look at this. If this is gated by voltage, how does this thing open and close? All right. What happens, we have to go back to here, is the voltage sensor, something must be able to detect a change in voltage. Okay. And that something is segment number 4. Segment number 4 has arginine and lysines every 3 or 4 amino acids. As you go across these 21 different residues, every 3rd or 4th is a positively charged residue. Okay, that's the voltage detector. So what's going to happen is, we'll go back to this picture. Okay, so this thing is closed, the gate's closed when we're at resting potential, which you know is -60, or -60 or -70. Negative on the inside, positive on the outside. Okay. If we depolarize this using a variety of mechanisms, okay, so we now depolarize, this goes from negative to positive. What effect do you think this positive charge is going to have on S4? Well,
Repellent?
pardon me?
Repellent?
Repellent, yeah. And what it actually does is it pushes it up and it turns a full 180 degrees. It does a 180 rotation as it turns. That opens the gate. So that's how that S4 works. It's sitting there minding its own business, and then this thing depolarizes and it pushes up on S4. S4 moves upward and turns. That pulls on to that opens the pore, and then the ions can go through. Really cool. Okay, so now we know how the voltage gating works. Go back here. We got to account for this thing. On the N-terminus of every single subunit is this little ball. Looks like a little ball. Okay, it's called the inactivating particle or domain. It's very close to the N-terminus. So how many inactivating particles do you have per channel? 4, because there's 4 subunits, 1 ball per channel. Okay. And some of you have probably played that little game where you have this little cup and you have the ball attached by string and you try to pitch it into the cup. That's what this thing is doing. It's trying to pitch that ball into the cup. Okay? And what makes it move is when the channel opens. Okay? It starts a slow movement, slow by channel, the channel clock. The channel clock works on millisecond timescales. So slow to it is like 10 milliseconds. So what's going to happen is this is off, and the channel is considered to be activated because the ball, none of the balls are blocking that pore. Okay. Now, as this thing opens, you have two types of changes that are going on. You're opening the channel, the pore, that is the S6 segment is now opening, allowing potassiums to move. But at the same time, it's like moving your hand with that little ball toy. As you move your hand, it's going to tug on this, and one of those four is going to try to get into there. Okay? But it's moving so slowly on millisecond timescales that we spurt through here a bunch of potassiums and do what we need to do before one of the little balls catches in the pore. Okay? And so it's the slow movement that was initiated by the opening is what causes the ball to swing in. And at the point that it blocks that pore, the channel is open, but it's inactivated. Okay? Now, eventually that channel is, the memory is going to repolarize. Okay? It'll go back to here, and the pore will close. That causes a conformational change that causes the ball to drop out. So now we're back to where we started. So we have open, we have closed and activated. We have open and activated. And the third state is open but inactivated. So those are the three states that go round and round and round. But what initiates it is this part right here, the depolarization. Okay. And once you understand how this kind of thing works, you now know how sodium, voltage-gated sodium channels work, voltage-gated calcium channels. They're all related in structure, but there are small differences among them. The voltage sensor might be in a different transmembrane segment. There might be different subunits, but we don't need to memorize all this. That's our poster child for a voltage-gated channel. Okay. Any questions? Yeah.
The amino group shown at the end of the chain,
Right.
That doesn't have anything to do with the mechanism at all? It's just N-terminus?
Well, except that it shows you where the ball is.
Okay.
That's why they show it. Yeah. But what's wrong with this?
It should be charged.
It should be positively charged, right? Okay, good. All right, let's take a break here. Unless there's more questions. Okay, why don't we go ahead and get back. Now, so if you can't use the Nernst potential to calculate, sorry, not Nernst, but the Michaelis-Menten to measure the kinetics of how fast this is going through, you must be able to use something, and they have figured that out. It's called the Goldman-Hodgkin-Katz equation, and it's this kind of complicated thing. And so if you take a physiology course, you get in there, but you can actually calculate the rate of ion flux through that channel as a function of concentration and membrane potential. So it's nonlinear, obviously. So, but we're not going to worry about that. Okay. So let's move on to. We've talked about voltage gated. We talked about a leak channel, which is the bacterial channel. We haven't talked about pardon me, a ligand gated channel. And the classic one is an acetylcholine activated channel. So the neurotransmitter actually is acetylcholine. And so this is a presynaptic nerve terminal. A neuron is coming down, and this is, you can see it here. This is the axon, and it splits up, and it makes contacts all along this little tube, and that's a muscle fiber. It's skeletal muscle in this case. So all your muscle fibers are running here, and so this thing is sending a signal to contract, 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 right? So if we go back to the cartoon, what's happening is here's this axon terminal, and you have these vesicles that are loaded with acetylcholine. Okay, so that's going to be the ligand. And so when you want this muscle to twitch, what you have to do is there needs to be an action potential, a nerve impulse, that is generated by both the voltage-gated sodium and voltage-gated potassium channels. And so there you got a wave of depolarization that reaches the axon terminal, and that depolarization wave opens the voltage-gated calcium channel. And so that opens in the plasma membrane, and a calcium goes in, and the calcium binds to specific proteins that we'll talk about in 2 weeks that cause this thing to fuse with the plasma membrane. And if that vesicle fuses with the plasma membrane, it unloads its contents into this little region called the synapse. And at this point, this is very close to that. So diffusion is fast enough to move that acetylcholine molecule onto this ion channel, which is called a nicotinic cholinergic ion channel. So the word cholinergic means that the neurotransmitter that activates it is acetylcholine. But you've got two flavors of cholinergic channels. You have nicotinic, which is the one we're going to study, and then you have muscarinic, which we'll look at later. So this is the poster child for our ligand-gated ion channel. And it is a cation channel, so let's blow that up and take a close look at it. And so it oscillates between three different states. It's unoccupied, so there's no acetylcholine. This is the muscle cell membrane. This is where you are, so don't get lost. You're not back in the neuron, right? And so this is the synapse up here. And then you're going to get a little squirt of acetylcholine that's going to bind to this protein, which is going to gate the channel open. And then after a delay, it will still be bound, but this will inactivate. It closes. Okay. Now let's look at the details. So this has 5 different subunits. So it's 2 alphas, a beta, a delta, and a gamma subunit. Okay. And the binding site for acetylcholine is at the junction between both of the alphas and the single beta. So it's right nestled in between alpha gamma and alpha gamma. There are 2 acetylcholine binding sites that are going to gate this channel. Okay? Now, you have 5 subunits, but only one of the alpha helices in each of these 5 different subunits is used. So we take a look at, this is an alpha subunit. One alpha subunit. Notice it's got 1, 2, 3, 4 segments, but then we don't call them S1, 2, 3. For whatever reason, probably because it's a muscle cell, we call it M1, 2, 3, 4. Which one of these lines the channel? M2. M2 is the alpha helix that is the selectivity filter. So that's this blue thing right here. And notice there are residues at the entrance to the cytoplasm and the exit to the synapse that have negative residues. What's that going to do to the selectivity? What kind of ion is going to go through here? Cation. So that's very good. Cation selective. So it has to have a selectivity, and then once it gets in here, the actual affinity or specificity is determined by other residues. Okay. All right. So the actual gate is controlled by several leucine residues, and those are hydrophobic residues that when they come together, they shut off the aqueous pore and occlude the channel. So that's where the gate is those leucine residues on M2 of all 5 of the different subunits. Okay. But actually what can go through here is 3 different ions. It's a cation selective, but it's not like the potassium channel. It will take sodium, potassium, or calcium. It's pretty sloppy, actually. But when this thing opens, one of those 3 is preferred to go through, and that's sodium. Okay? Then you should be asking yourself, why does it choose sodium over potassium and calcium? It can take all 3 of them. Not at the same time, but I want you to think about that, take that home as a homework problem. Why does it select sodium going through here? That's actually fairly complicated. I think it might be answered in your book, but I'm not sure. Okay, so why is it that sodium goes through here? Well, one thing you can say about sodium is there an inwardly directed electrochemical gradient? What's the concentration of sodium on the inside of the cell? Low or high? So you need to know that. In all cells, because of the sodium pump, sodium is low on the inside. Okay, and what's it out in the bloodstream? It's 150 or something. So it's 150 out, and it could be 5 or 10 millimolar inside. So there's a chemical potential to go in. And is there an electric chemical potential to make it go in? Yeah, when this thing opens, before it depolarizes, it's minus 60 or 70. Sodium loves negative charges, and so it's going to be sucked in here by pushed in by the chemical potential and pulled in by the electrical potential. There's always 2 forms of energy if it's charged solute.
But that's the case for calcium too, so that can't be the whole picture.
That can't be the whole picture. Right. Okay, and potassium, remember, even though the chemical potential is to go out, so why doesn't potassium go out? So that's a tricky one. All right. Okay. So I think where that leaves, are there any questions before we leave the nicotinic? So this is a cholinergic nicotinic ion channel that's ligand gated. Mouthful, right? And one of the reasons it's called nicotinic is that nicotine is an agonist for this channel. You put nicotine on here, that will fit right in there. Open. Okay. Okay, so the last little remaining thing is to, number one, understand the basis for a membrane potential, because it just confuses the heck out of a lot of people. And then the second thing is, if you've got competing directions, like you're trying to figure out which way an ion is going to move through the channel, and the chemical potential is pushing it in, and the electrical potential is pushing it out, how do you know which way it's going to move? Well, to do that, you have to solve what's called the Nernst equation. Okay. So, but let's first let's go back to Toronto, all right, and bring their website up again. They have a really, and you can go through this as slowly or as quickly as you want to. Yeah, resting potential, is that it? No, yeah, let's do this. Okay, so let's start at the simplest where there's no membrane potential and ask the question, what causes it? Okay, it's got to be an unequal distribution of charge. And so something has to move those ions across there, so it's got to be a transporter or a pump or an ion channel. Okay, so originally it starts at 0, 0, okay, and we're gonna change that though. Okay, so here we are. We've got a resting ion channel, right? We've got an ion pump, and so all of these things are present and they're cooperating in order to make this thing work. Okay. So the first way we can create an asymmetric distribution of ions is to use ATP to pump something out. So if we do that with the sodium pump, let's make it work. Do something. Oh, here we go. It's going to do its thing. It's going to pump. And notice you just put 3 out and you put 2 on the inside. So what's happening? You're starting to create a membrane potential, aren't you? Because you're putting more positive outside and less positive on the inside. So you're already starting. This needle should move there in a minute. Okay, so it becomes more positive on the outside. Okay, what's that going to do to things on the inside? It's going to attract negative charges to the membrane. So this is really thin, so it can actually store a little charge. That property is called capacitance, but you don't need to know that. But this has a unique electrical, it can store a lot of charge, okay, a membrane potential. And so we got the excess sodium out there because of this, we pumped it out. But what actually is attracted to the membrane, the cytoplasm is full of anions, phosphate groups, sulfate groups, proteins, nucleic acids. They're all negatively charged. So we have an excess of negative charges to play with. That's really the basis for most of the membrane potential, or part of it. Okay, so you can already see this thing actually should be moving. So yeah, it moved from 0. Now it's hyperpolarizing, or it's becoming more polar, I should say. So now you're starting to see why it's negative on the inside. But now, what if we add a resting K channel here, okay, and we allow it to operate? Okay, there's actually a chemical potential that's pushing it out, but it's only going to go through there until the positive charges get strong enough to block it from going out, right? And the point at which that happens is the equilibrium potential for potassium. So if we can build up enough positive charges, that will block the potassium from leaving. That's called, the membrane potential at which that thing happens is called an equilibrium potential, because this is now at equilibrium. It means, I think they show it here, K flows out, ions continue, they'll keep going, okay? And notice we've reached like minus 90 over here, right? That's the equilibrium potential for potassium. That's the point. That's a fancy way of just saying, I think they show it, the net movement of potassium stops. It goes in, oh, we hit minus 90, now they start to come back in. And it doesn't mean that the ions stop moving, it means there's no net movement. It means for every one that goes out, there's one that comes back in. That's the equilibrium potential. And for potassium, it's different for every single ion. And for potassium, based on the concentrations, it's minus 90. Okay. And that's what we'll use the Nernst potential, the Nernst equation to calculate. We can calculate the equilibrium potential for any ion that's going to move. Okay. So now, but what's our typical Joe Blow membrane potential? So why is it more depolarized than that? So this is minus 90. Something must push it back in the other direction. Well, that something is there's a few sodium ions, sodium lead channels. There's not very many of them, but when they open, which way does the sodium go? It has to go in. That's what pushes the minus 90 back towards minus 70 or 60. So it's the operation of two open channels that's doing that. They're fighting. The one that wins is the potassium channel because there's more of them. And the way we say that is there's a higher permeability to potassium than there is to sodium. If there were more sodium channel, this needle would go way past this and would go close to plus 60 because that's the equilibrium potential for sodium. So is this clear what's causing this? First you pump it out, you use energy, then you attract anions to the membrane, okay? And then you open channels and they move in response to this little, and they generate a large electrical change based on the Nernst potential. So all of those are cooperating, and you can just step through this if you don't get every part of it. But do we have questions before I leave this? Okay. And this whole process would stop because if there were no ATPA sodium pump, the sodium would eventually depolarize the cell back to zero, right? But the pump keeps operating, right? And so you have all things operating simultaneously. The net result is the membrane potential, which is a steady state. It's not at equilibrium. It's displaced from the potassium channel equilibrium and the sodium channel equilibrium. So we're at a steady state because of all these things working together. Okay. Good. So lots of things moving around here. Now, if that's all we could do with it, it wouldn't be very useful. So what we're going to do now is go back to the PowerPoint. And notice we have all these situations where it's really clear to predict which way the ion's moving. This is the difficult one, where the chemical potential is pointed in and the electrical potential is opposing. Who wins? Well, I just told you how you figure that out. Is you got to be able to calculate the membrane potential that will exactly counterbalance that chemical difference. That's the Nernst potential. And you can calculate that for every single ion. So let's do that, okay? And you can do this exercise at home. We'll do a couple of examples, but these are the basic charge distributions. And so you can look at them and come up with problems and calculate the Nernst potential for every single one of these ions based on these concentrations. So what do you have to do? What do you have to know in order to utilize the Nernst equation? Let's get to that equation. And we'll start out with the so-called complicated version of it, and then we'll simplify it. So okay. And let's get a little bit bigger. We can still go bigger. There we go. I think you might be able to see that. Okay, so this is the full Nernst equation in all its glory, and it's like the inside membrane, the inside voltage versus the outside equals this thing. Okay. And what we can do is simplify this by, the outside voltage is always taken to be 0, so it's really what the voltage is inside, right? So we never deal with this thing, we just deal with the voltage inside the cell with respect to the outside, which is 0, okay? So this is the membrane voltage at which the ions, this solute S, stops moving in a net direction. It's moving, but it's not moving more outside than it is inside. It's just going back and forth. Okay, that's at equilibrium. So that's what this thing is going to calculate. And notice it's a function. It has all these constants which you can look up and amuse yourself with, but there's just a few variables. One is temperature. Temperature actually, it says a weak dependence on temperature. So for a given temperature, you need to know what that value is. Okay. You need to know the valence on the ion that's moving. So whatever solute is, so if it's sodium, the valence is +1. It has a sign and a magnitude. Okay, it's got a sign and a magnitude. So for calcium, the valence is +2. Chloride is -1. But you have to put that valence into the equation. Okay, and then all you need to know is the concentrations inside and outside the cell. And you need to know the ratio out over in. Just ingrain that into your head. It's out over in, out over in. And you've got this thing solved. All you have to do is plug that ratio in here, take the log, okay? So let's say if it's 10 times more concentrated outside than in, it's 10. A log of 10 is 1, okay? And you multiply it, that log value, which is 1, times these numbers. At 20 degrees room temperature, at which these old scientists used to conduct all their experiments, mostly with squid axons, room temperature was 20. So at 20 degrees, it's 58. All those constants, so we can just get rid of all this 2.303. That's the conversion from natural to base 10 log. It's 58 millivolts at that temperature. At body temperature, it goes up to, wow, 61.5. So it's a different value. But this is a very simple equation. But knowing the equation does not allow you to answer the question, which is which way is the ion going to move. You have to be able to get this number and then use it. Those are two different things. So it's no good just to plug it into your calculator. Do get out some of you get out your calculators though. I'm going to ask somebody because I don't want to operate a calculator and do this too. So let's do a real simple example, and we'll do a case where you know what the result's going to be. Okay, let's do it for sodium, because you know if you open a sodium channel, it must go in because the chemical difference is pushing it in, and the electrical difference is sucking it into the negative inside the cell. Okay, so I suggest you always draw a little cell and put the sodium concentration out. Let's get some numbers here. Here we go. So 145 millimolar outside, and so inside the cell you know it's going to be lower, so it's 10 millimolar sodium inside, right? The membrane potential, and remember it's Vn, okay? With respect, what's the outside? 0, okay? So it's -60, okay? And we're going to open a channel. If there's no channel that's open, which way is the sodium going to go? In or out? That's not a trick question. It's an important distinction. If there's no pathway for the ion, can it go across the lipid bilayer? No, there'll be no movement. You have to have an open channel, and it has to be just sodium channels. It complicates the situation to open a chloride, potassium, and a sodium channel at the same time. Can we do that calculation? Yes, take a physiology course and learn to do that. But that's not what we're doing here, okay? So we're going to open this and ask the question which way it's going in. You know it goes in, okay? So let's solve for the equilibrium potential value, the E sub sodium, right? And let's do it at 20 degrees room temperature. So we have 58 millivolts times our the log. So it's 58 divided by the valence. What's the valence? A plus and a 1. Okay, so it's +1. Okay, times the log of, is it in over out or out over in? Out over in, out over in, out over in. Okay, so out is 145. So somebody take the ratio of 145 divided by 10. Well, you can do that. It's 14.5, right? Take the ratio and then take the log. Don't do the other way around. So do the ratio. What is the log of 14.5? Somebody do that.
1.16.
What was it?
1.16.
1.16. So we're going to multiply that times 58 and a plus 1. Okay, just keeping that there. And what's the product of that? What is it? Somebody?
57.36.
57?
No, 67.
Let's just say 67. That's close enough. Okay, 67 millivolts. So what we've just done is convert millimolar concentration into an electrical potential. We're going to see if they push, which way they push the ion. Okay. So this doesn't solve the problem. You need to do this one last step. Is put yourself a number line down here with a 0. It's a unidirectional. There's no y-axis. Okay. And so label on this axis the Nernst potential for sodium of the ion that you just calculated. And so we're going to go over here. We're going to go ENA is plus 67 millivolts. So it's way positive. Where's the membrane potential? It's down here. I said, what, minus 60? And you identify that, right? Okay, so here's the rule. When you open that channel, the membrane potential is going to collapse towards the equilibrium potential. That means that there's going to be pressure to make sodium go in or out as long as these two are not equal to each other. And so as sodium starts to go into the cell, the membrane, so you tell me if this is what's happening. So the sodium is here and you open this. If this is going to move more positive, which way must sodium go in to make the interior more positive? In or out? Yeah. It's actually going to go like this. And so this is going to go that way. But the rule is the membrane potential always moves towards the Nernst potential, and all you have to do is figure out which way that ion that you're dealing with needs to go in or out to push it that way. In fact, this difference in energy is the electromotive force on the sodium ion. It's called EMF for sodium. And it's equal to the membrane potential minus the Nernst potential. It's a really simple, it's the difference between those two little goalposts. And the more sodium goes in, the more energy you dissipate until you've used it all up. Because there's nothing adding energy, it's just the gradient that's being dissipated that's moving the ions. Okay? All right, so try this at home with all the ions in that table and see if you can do this.